Hi guys, so my name is Caitlin and today I will be presenting Emily's a Nice project. So what exactly is immunological tolerance? Immunological tolerance is the lack of response to antigens by exposure of lymphocytes to these antigens. Tolerance is maintained by via and peripheral tolerance. So what exactly are central and peripheral tolerance? Central tolerance is immature lymphocytes that recognize self-antigens, which may come across these antigens within the generative lymphoid organs and are subsequently eliminated. B lymphocytes undergo a shift in their specificity through receptor editing, while certain T lymphocytes mature into regulatory cells. Peripheral tolerance are fully developed self-reactive lymphocytes, which might undergo inactivation or elimination upon encountering self-antigens in peripheral tissues, or they could also be subdued by regulatory T cells. So central mechanisms. There are three mechanisms that take place in central tolerance. The first one being receptor editing, which is certain immature B cells detect self-antigens in the bone marrows, which might reactivate RAG genes, recommends Ig light chain gene recombination, and subsequently manifest a new Ig light chain. The second is deletion, which takes place when editing proves to be inefficient. The immature B cells that strongly identify self-antigens receive signals for apoptosis and perish accordingly. The last one is energy. Certain self-antigens may be identified in the bone marrow with low avidity. B cells targeting these antigens may survive with reduced antigen receptor expressions, leading to functional unresponsiveness. So peripheral, there's also three main mechanisms. The first one being energy. When T cells identify antigens without co-stimulation, the TCR complex might lose its capacity to convey activating signals. On recognition of self-antigens, T cells may also show a preference for engaging one of the inhibitory receptors in the CD28 family. The next one is suppression. Regulatory T cells form either in the thymus or peripheral tissues upon encountering self-antigens, aiming to dampen the activation of potentially detrimental lymphocytes targeting these self-antigens. They achieve immune suppression through various means, which are releasing cytokines that hinder lymphocytes, dendritic cells, and macrophage activation. The last one being deletion, a decrease in intracellular anti-apoptotic proteins alongside an increase in apoptotic proteins, or alternatively, the upregulation of death receptors and their corresponding link ligands like FSA and FSA link ligand. What is autoimmunity? Autoimmunity is an immune system's response against self-antigens. Genetic susceptibility and environmental stimuli can lead to autoimmunity. So what is genetic susceptibility? Different genetic loci may contribute to the susceptibility to autoimmunity, potentially by impacting the regulation of self-tolerance. In life, many autoimmune diseases in humans are linked to particular MHC alleles. Variations in non-HLA genes are linked to different autoimmune conditions and might play a role in the breakdown of self-tolerance or the abnormal activation of lymphocytes. Environmental stimuli. So infections and other inflammatory triggers facilitate the migration of lymphocytes into tissues and the activation of antigen-presenting cells, subsequently leading to the activation of self-reactive T cells and consequent tissue damage. Microbes can also stimulate APCs to express co-stimulatory molecules. When these APCs present self-antigens, they activate specific T cells instead of inducing self-tolerance. Certain myobasal antigens might exhibit cross-reactivity with self-antigens, a phenomenon known as mimicry. Consequently, immature immune responses triggered by these microbes may target self-cells and tissues. Hypersensitivity. Hypersensitivity reactions are classified on the basis of the principal immunological mechanism that is responsible for tissue injury and disease. So there are four different types of hypersensitivity. The first one being immediate hypersensitivity, which is a pathological reaction triggered by the release of mediators from mast cells. Type 2, antibodies targeting cells or tissue antigens can lead to the damage or dysfunction of these cells or tissues. Type 3, Antibodies against soluble antigens have the potential to form complexes with these antigens. These immune complexes may then accumulate in blood vessels across various tissues, triggering inflammation and tissue damage. Type 4. Delayed type hypersensitivity involves diseases stemming from the responses of T lymphocytes frequently targeting self-antigens within tissues. So the mechanism of type 1, it is usually in individuals who are prone to allergies. Exposure to some antigens result in the activation of Th2 cells and the production of IgE antibodies. The IgE antibodies bind to high affinity Fc receptors specific for the heavy chain that are expressed on mast cells. These mast cells are exposed to the allergen that are activated to secrete their mediators. Phases of type 1. Immediate hypersensitivity response is separated into the immediate reaction, which usually occurs into 1 hour, and the late phase reaction, which occurs 2 to 24 hours after. Morphology of the immediate reaction is characterized by vasodilation, congestion, and edema. Late phase reaction is characterized by inflammatory and fillet-rich in eosinophils, neutrophils, and T cells. Mechanism of type 2. Antibodies specific for cells and tissues antigens may deposit in tissues and cause injury by inflammation, optimization, phagocytosis, and abnormal cellular response. Mechanism of type 3. Large complexes can be cleared by macrophages, but small immune complexes cannot be cleared, and they insert themselves into small blood vessel joints and glomeruli, causing symptoms. And the last one, mechanisms of type 4. Major causes are autoimmunity and exaggerated or persistent responses to microbicidal or other environmental antigens. Against environmental antigens, including contact sensitivity to chemicals such as poison ivy. Tissue injuries also may accompany T cell responses to microbes. Superantigens, which are certain microbicidal toxins produced by some bacteria and viruses, can lead to the production of large amount of inflammatory cytokines, causing a syndrome similar to septic shock. And that is the end of our presentation. Thank you.